I'm going to just shift gears now and talk a little bit about genotyping technology, but really the message that I want to leave you with is that genotyping technology is changing rapidly, and that markers really aren't the limitation anymore. Okay, what this slide is illustrating is the scheme for creating next generation sequencing. So you create a library, that library goes through some quality control, it then gets sequenced in a flow cell. That sequence data is basically image intense, and that image data is then deconvoluted. You go through an assembly process, and you've got data that can then be mined for polymorphisms. Okay, so in the context of SoulCap, we mine that data for polymorphisms for the Illumina array, but sometime in the very near future, you're going to do your SNP calling based on this kind of data. Or more importantly, somebody's going to do it for you, right? Okay. You know, what I'm going to do here is just go through really some of the statistics on the tomato chip, and I want you to see how well they parallel the potato chip. So again, we were working with a 10,000 probe array. We had 8,784 SNPs. So some of those SNPs had to use 2 probes to score. 7,627 passed quality control. We cross-validated with the INRA and the trait genomics data set. There were community candidate genes, and that many of these SNPs work within cultivated populations. It also turns out that they work very well with wild populations, and I know that was a question, but over 3,700 of these SNPs have been mapped in a reference population that's cultivated by wild species. Okay, so it's like in potato, the tool is working both for cultivated as well as wild species. I guess the other thing that I want to leave you with is that, again, because of the pipelines that Robin's group developed, and because of some other tricks that we use in terms of using allele frequencies in making decisions about our content, we had a very, very high validation rate. You know, essentially overall the validation rate was over 97%. Okay, so it's a very robust set of tools. And then if we start to look at pairwise comparisons, there are simply a lot of SNPs available even for biparental crosses, and that's really what the theme of this slide is. So you can look at the pairwise comparisons and see that there's a lot of markers to choose from. So these Illumina arrays are excellent survey tools. And in terms of how one would use a tool like this, I think what we need to do is give a lot of thought to the germplasm that we're going to genotype. Is that going to represent a regional breeding program, like all of the Northeast for potatoes, or is it going to represent your personal breeding program? Right? We have to think about what comparisons we want to make, which is influenced by the questions we want to ask of the data. So in the case of potato, you might want to ask if, I know that many of the potato breeders run programs that create material for both fresh market and processing, right? So you might want to ask whether you actually have differences in those germplasm classes. Do you have structure that correlates with those different aspects of your program? And then finally, really the goal is to ask what sequence variation is important to the phenotypic variation that we see. Okay. Another bit of information that can come out of these survey tools is that we can begin to look at the distribution of variation along chromosomes. And what this slide is showing is just 2 chromosomes for tomato, chromosome 1 and chromosome 2. There are 3 germplasm classes illustrated here. Vintage material, which is what we might call heirlooms and land races, fresh market material, and processing material. And what you can see is that there is differentiation between the germplasm classes. So there are portions of chromosome 2 that remain polymorphic in processing tomato, but have been completely fixed in the fresh market germplasm that we looked at. Okay. These are the kinds of features that you're going to see that represent structure that are inherent in either market class breeding or even regional adaptation. Okay. Now, another thing that you're going to see is population structure or subdivision that is based on some aspect of selection. Okay, and so when we run a Bayesian analysis using the program called structure, what we see is that across the tomato germplasm that we genotyped, we see at least 6 distinct subpopulations. So the land races classify in a group, the vintage classify in a group, the fresh market and the processing are distinct, and even within the processing there appear to be distinct substructures, right? And these correlate with regional breeding programs. Okay, so California material represents a distinct subpopulation from Midwestern material, right? That may reflect adaptation, so selection for specific environments, or it may simply be residual from founder parents, right? We don't know that yet. I do want to point out that this analysis has already been done in a preliminary way for potato, and so if you look at the paper that was published out of Robin and Dave's work in BMC Genomics, and the link is here, you'll see that they've run this analysis and that you do see at least the chipping types in their own subpopulation relative to other potato types. Okay. Now there's a reason, I'll come back to structure, but there's a reason that knowing the structure of your subpopulations is important when it comes down to association analysis and genome-wide selection, and I'll follow up on that in just a little bit. Another thing that you can get out of this kind of genome-wide survey analysis is an estimate of how linkage disequilibrium decays. Okay, and this is important because it really tells you what the important marker density is for your crop. So in outbreeding species like maize, LD can decay within 1 kb. Okay. In inbreeding species like tomato, that's not the case. And in clonally propagated material like potato, it's also unlikely to be the case, right? You make a cross, you have one opportunity for recombination, but you've got whole parts of the genome that just don't recombine because your population isn't large enough. Okay? And so the bottom line is that in tomato, LD decays over like a 5 centimorgan window, right? So we don't need high marker density to capture a lot of information, all right? So that's what's demonstrated on the top of the slide for both processing and fresh market material. Now another important thing that we see within processing tomato, and this is really reflected in the subpopulations we see with the structure analysis, is that we have significant gametic phase disequilibrium. Now that's a big phrase right there, but really what it means is that there are genes on different chromosomes that appear to be linked because of selection. Okay? And really that's what we do as plant breeders, right? We are selecting for combinations of genes across the genome. Okay? We can see this in a very striking way with tomato. Okay? So there are whole sections of the genome that are essentially co-inherited, right? Now, that actually has a very negative implication for our ability to use association mapping, right? And I'll explain why in just a little bit. Okay. First, I want to talk a little bit about some strategies in terms of genotyping breeding populations. So one of the things that was implied in some of what Dave was saying in his talk, and is also very clearly seen in the tomato material, is that we may not need to use that high-density array for all applications. In fact, we probably don't. So what I'm showing on this slide is a table on a chromosome-by-chromosome basis on the number of polymorphic SNPs in selected tomato populations. So this particular set here is for processing tomatoes, not fresh market. And what you can see is that if we look at chromosome 1, there's only really 75 polymorphic markers on that chromosome. But if we then impose some kind of a filter and say, look, I don't want to see markers that are closer than half a megabase, that number drops down to 20. Because some of the markers are clustered. And if instead of using physical distance, we were to use recombination in a mapping population, what we would see is that a lot of the markers that are closer than 1 megabase simply never recombine because the populations aren't large enough. Okay? So if the markers don't recombine in our populations, they're not giving us new information, but we're still paying for that data point. Okay? So the point that I want to leave you with on this slide is that we can take these 7,600 SNP arrays and we can reduce it to a set of markers that are informative, highly informative to our populations. And then the next step is to decide how you're going to use it. Are you going to use it to facilitate some sort of a backcross program? So if you want to eliminate wild species genetic background in an integration program, you could maybe get away with just 48 SNPs that cover the genome, right? If it's really for genome-wide selection, you maybe want to go up to 384 or higher, depending on the population size, right? But the point is that we can create subgroups of SNPs and much more cost-effectively do what we need to do. Okay, so the Infinium array is an excellent survey tool to look at program diversity and substructure. If I were to make recommendations on how to use that survey tool, I would say make sure that all the germplasm in your particular program at the point where you start to collect phenotypic data gets genotyped with that tool. You can use that for possibly for association analysis. We'll put some caveats on that. You can use that survey to develop smaller, more cost-effective subsets of markers for subsequent selection and background genome elimination. And you can use that smaller set for very effective mapping in early generation material. Okay, with the number of markers really just dependent on the population size and your selection goals. Okay, and the other thing I want to point out is that in SoulCap we've relied heavily on Illumina, but there are other companies. There's K Biosciences, for example, is a company where you can order a large number of primers. They can store those primers for you. You can then select subsets of them to run on material, and they basically are going to do it on a per sample basis. You know, TACMAN reagents can be purchased in this way as well. And so a lot of this would be worked out based on maybe a core facility that you have access to or a service provider that you elect to use on the project.